So we left off with the real overall summary equation shown at the bottom of this slide, C6H12O6 plus 6O2 plus 30-ish ADP and 30-ish PI, inorganic phosphate, yields 6CO2 plus 6H2O plus 30-ish ATP. And so you may have instructors that give exact numbers for those blanks, but research has shown that we don't know that precisely. So something in the 30s is good enough for me. We need to know the mitochondrial structure. The mitochondrion has an outer membrane, and this picture has a cutaway, so you can see that in kind of outlined in red. And then there's a space inside the outer membrane, but not inside the innermost membrane. And that space is called the intermembrane space. And then you have a membrane that's heavily folded on the inside. The inner membrane is called the inner membrane. It's very heavily folded, and it's outlined here in yellow. And then in blue, you have this space called the matrix. And you may recall from endosymbiont theory or endosymbiosis that within the matrix or within the mitochondrion, there will be DNA molecules and ribosomes, and you can see those. The artist is trying to show that. Not that well, but mitochondria have chromosomes and ribosomes. So anyway, but we're focusing on the membranes and the spaces. So outer membrane, inner membrane, heavily folded. The matrix is the space inside the inner membrane. The intermembrane space is the space between the outer membrane and the inner membrane. Okay, so you're going to want to be able to label parts of the mitochondrion. Now, moving on, the stages of aerobic respiration. So, or we sometimes call it the oxidation of glucose. We start with glycolysis, pyruvate oxidation, citric acid cycle, electron transport chain chemiosmosis. So that's 1, 2, 3, 4. I've color-coded each of these, so on the next slides, as we're talking about each one, the title will be color-coded. So you can kind of keep it in chronological order. This slide shows the spatial organization, in other words, the location of each of these 4 stages with regard to the mitochondrion. So glycolysis happens in the cytosol, in the cytoplasm, so not inside the mitochondrion itself, so just in the cytosol. So that's why it's shown here outside of the mitochondrion. The other 3 stages, stages 2, 3, and 4, pyruvate oxidation, citric acid cycle, and electron transport chemiosmosis all happen inside the mitochondrion. And we will be a little more specific as we focus on each of these 4 stages moving forward. So glycolysis is a 10-step biochemical pathway. So it involves 10 different enzymes that act in sequence, starting with a very starting reactant molecule and ending with a particular product molecule. It occurs, like I said, in the cytosol, in the cytoplasm. The initial very initial material that it starts with is glucose, 1-glucose, so C6H12O6. And the final product of glycolysis, just glycolysis, is 2 molecules called pyruvate. Glucose has 6 carbons. Each of the 2 pyruvates contain 3 carbons. So fundamentally, what you have in glycolysis is just the splitting of glucose in half. In fact, the name glycolysis means sugar splitting. Ultimately, we produce 2 ATP molecules in glycolysis. We say 2 net ATP molecules. And the word net means that there's going to be an investment or a using of 2 ATP molecules to start the process. You have to put some energy in, but you get a total gross yield of 4 ATPs. So the overall yield of gross profit minus investment is 2 net ATPs. if we think about it almost in a business sense. And then we also get 2 NADHs. Remember, NAD plus is our electron carrier. So 2 of those pick up electrons, and we get 2 NADH molecules. This is the overview of glycolysis. You do not have to memorize this, but it gives you some sense of the steps. Even this picture doesn't show you all 10 steps, so you can find even more detailed diagrams of each of the 10 steps. We don't really need to go into that level of detail. What we see here is the 6-carbon glucose starting the process. At the bottom, we end with 2 molecules of 3-carbon compound called pyruvate. You can see a few places where ATP is shown as a reactant and 4 places where ATP is shown as a product. Remember that reactants are always on the stick side of an arrow, and products are always on the pointy side of the arrow. And so if you follow that, you can kind of see that happening there. Again, this is an artist's depiction of what's happening. Now, what happens to pyruvate? If oxygen is available, then pyruvate will move on to stages 2, 3, and 4 of the aerobic respiration that I'm teaching you right now. However, if there is no oxygen present, either fermentation will happen or, I don't know, I didn't list anaerobic respiration. Usually fermentation is going to be kind of the backup. So at the moment, so glycolysis can happen no matter if there's oxygen or not. But then that point becomes the decision-making point. So here's the production of pyruvate from glycolysis, and then there's a bifurcation of sort of the decision tree. If the cell has oxygen, then it'll move through the remainder of the aerobic respiration stages. If it does not have oxygen, it will typically go into one of the fermentation processes. So we're going to focus on this left-hand side here. Aerobic respiration, there is oxygen, and so we'll continue on in that direction. So in pyruvate oxidation, which is stage 2, the pyruvate has to be moved into the mitochondrion. It's actually moved into the matrix part of the mitochondrion. And in the matrix of the mitochondrion, pyruvate is going to be oxidized, again, broken down even more. And remember, each pyruvate has 3 carbons. So what's going to happen is 1 carbon is going to be broken off, okay? And when 1 carbon is broken off and released as carbon dioxide, 2 carbons remain, and they're bonded to each other. So the 1 carbon is released as carbon dioxide, and the 2 carbons that are left are called acetyl. Now, this happens, like I said, in the mitochondria. In a prokaryotic cell, this can happen at near the inner side of the plasma membrane. So here's the reaction shown sort of in a vertical fashion. Here, 1 pyruvate is shown, and then this down arrow is the overall reaction. 1 carbon dioxide is broken off. 1 NADH is produced, because it picks up those electrons from that covalent bond, and then also produced is an acetyl. But acetyl it needs something to bond to right at that moment, so it binds to a coenzyme that's hanging around called coenzyme A. So, but it's really the acetyl part that we're interested in here. Now, since there are actually 2 pyruvates that were generated in glycolysis, we really have 2 pyruvates coming in, 2 CO2s being released, 2 NADHs being released, and 2 acetyl coenzyme A's being produced. We're going to summarize this on a chart at the end of this segment. So a little fun fact, arsenic is poisonous because it is an allosteric inhibitor of the enzyme that catalyzes this particular reaction, which the enzyme is called pyruvate dehydrogenase. So arsenic is an allosteric inhibitor of that enzyme. So if it blocks, and any inhibitor that blocks an enzyme of respiration is typically going to be fatal. It's going to be poison. So like I said, for each 3-carbon pyruvate molecule, we get 1 carbon dioxide. When a carbon dioxide is broken off of a molecule, that's called a decarboxylation reaction, removal of a carboxyl, COO. 1 NADH is produced from 1 NAD+, and 1 acetyl-CoA is produced. Then we move to the citric acid cycle. This is the first time we've seen something called a cycle. A cycle occurs when you have a molecule already existing, goes through a series of steps, and by the end of the series, the same starting molecule is regenerated again. So what is at least one of the reactants has to also be at least one of the products of the series. If that occurs, then we call that a cycle. In the citric acid cycle, we have a 4-carbon molecule called oxaloacetate. And oxaloacetate is the molecule that serves as one of the reactants and one of the products. And so oxaloacetate is joined to the acetyl group that comes from the last step of the pyruvate oxidation. And when those two things are joined together, it makes a 6-carbon molecule. And that particular 6-carbon molecule is called citrate or citric acid, which is how this cycle gets its name, the citric acid cycle. This cycle is also, though, named the Krebs cycle after Dr. Krebs, who discovered it. So citric acid cycle or Krebs cycle means the same thing. Now, through a series of enzymatic reactions, the oxaloacetate is regenerated. 2 carbon dioxides are released, 1 ATP is released, and 3 NADHs are made, and 1 FADH2 is made. But you'll remember that there are actually 2 CO2s coming in. So whatever happens in this picture for 1 CO2, we really multiply it by 2. So we double it. So for each 1 acetyl-CoA entering the citric acid cycle, we get 2 molecules of CO2 released, 3 NAD pluses are converted to NADH, 1 FAD, which is a different kind of electron carrier but does the same kind of job as NAD, becomes FADH2, and 1 ATP is produced directly. And we say that the oxaloacetate, that 4-carbon molecule, is regenerated. So it's both a reactant and a product. This picture shows the full 9 steps, which you do not need to know, but each of these is catalyzed by a different enzyme, which potentially has different inhibitors that can block each of these enzymes. So up to this point, but by this I mean glycolysis, pyruvate oxidation, and the citric acid cycle, we have broken down the glucose completely into 6 CO2s. We have seen 4 ATPs directly made. We have a whole bunch of electron carriers hanging around, and those are the NADHs and the FADH2s. So what we're going to do, I'm going to have to start a very short, actually, I think I'll just fill this in and post it up for you guys. The summary of the first 3 stages that we've gone over, and you'll copy this down into your notes.